This is the third part of Module 24, the chemistry of life, organic and biological chemistry. By the end of this module, you will be able to recognize the amino acids and understand how they form peptides and proteins via amide bond formation. Also, you will understand the differences among the primary, secondary, tertiary, and quaternary structures of proteins. You will be able to explain the difference between the alpha helix and the beta sheet peptide and protein structure. You will distinguish between starch and cellulose structures. You also will classify molecules as saccharides or lipids based on their structures. You will be able to explain the difference between a saturated and unsaturated fat. Also, you will be able to explain the structure of nucleic acids and the role played by complementary bases in DNA replications. So let's start by talking about biomolecules. First of all, biopolymers are large biological molecules built from small molecules. Some of the examples of these biopolymers are the proteins, the polysaccharides or carbohydrates, and the nucleic acids. The lipids are large biomolecules, but they are not polymers. So let's start with the amino acids and the proteins. Amino acids have amine and carboxylic acid functional groups. They have both in the same molecule. Proteins are polymers of alpha amino acids, so that means that the amino acids are like blocks that you can combine to produce a protein. A condensation reaction between the amine end of one amino acid and the acid end of another produces a peptide bond, which is an amide linkage. So remember that one amino acid has both functional group. In one end, it has an amine, and in the other end has a carboxylic acid. So if you have two amino acid, one amine could react with the carboxylic of the other one, producing that peptide bond. So here we have, for example, this is an amino acid. We have the carboxylic here, and we have here the, actually this is the peptide bond, sorry. This is the amide bond that has a result from the reaction of the carboxylic acid of this amino acid that is in the left and this in the right. So we have a combination of two amino acids here. They produce a peptide bond of the carboxylic acid of one amino acid and the amine of the other amino acid. So for example, here we have the aspartic acid and the phenylalanine. We have two amino acids here and they create a peptide bond. You can see that they have an amine here, this amino acid, and this was a carboxylic acid, okay? And also here it has a carboxylic acid and the amine. So here we have one amino acid, another amino acid, and they react, they do a condensation reaction and create a peptide bond between the carboxylic of this amine and of this amino acid and the amine of this amino acid. So here we have a list of the natural alpha amino acids. They are really important in our daily life because they create proteins. A combination of this amino acids are the ones responsible to create the proteins. And as you can see, they have like a common structure. Okay, the amine, the carboxylic, and hydrogen. All of them have the same structure, but they have a different R group. Okay, so the substituent here, this is the most simple, the glycine with one hydrogen, alanine. This is the name. Also, we have different codes. We can use the 3-letter or just 1-letter code to represent an amino acid, valine, leucine. So we have different type of amino acids, and as you can see, they have this general structure here: the amine, carbon, the carboxylate, and the hydrogen, and then the derivatives that create different amino acids, the different derivatives. So let's talk about the protein structures. The primary structure is the sequence of amino acids in the polypeptide protein chain. So that combination of amino acid is known as the primary structure. The secondary structure is the interaction between the chain atom using the carbonyl and the NH atoms to produce basically a hydrogen bond, one hydrogen, and we have here lone pairs that give structures to protein. That's the secondary structure. The tertiary structure is the intermolecular forces between side chains atoms that give structure to proteins. And the fourth one is the quaternary structure. This arrangement of multiple units and or incorporation of non-amino acids portion of proteins. Basically, these are the arrangement of multiple units of those tertiary structures. So basically, this is a big picture of what we have mentioned before. This is the primary structure. We have here all the combination of the amino acids. And then this will turn to create the secondary structures, the interaction between the carbonyl groups and also the hydrogen of the amine of that peptide bond, and can create two different structures. We're going to talk a little bit of them in a few slides, the alpha helix and the beta sheets. And then they can interact the R groups from the amino acid between them, between an alpha helix and an alpha helix, or a beta sheet with an alpha helix, or beta sheet with beta sheet. And those interaction between those side chains are the one that create the tertiary structure, and then this tertiary structure can interact with other tertiary structure to create the quaternary structures. So let's go a little bit deep in each of them. The primary structure, formation of the amide bond between amino acid, as we mentioned before, and this repeat many times for a polypeptide protein chain. So here we have, as I mentioned, the glycine. We have an amino acid here, carbon, two hydrogen, because this is the glycine. We have one hydrogen here, the amine, and also the carboxylic here. And this is the alanine. So the combination of this carboxylic with this amino acid will create the peptide bond that we can see here, okay? And the production of water, okay? This condensation produces water. And here we have dipeptide, because we have two amino acid. And this can now interact with another amino acid and create another larger chain, and keep going until it creates the protein of that primary structure of interest. So here we have that combination. We have the nitrogen here, okay, the carbonyl. This is the peptide bond. There's the peptide bond here, peptide bond, the nitrogen and the carbonyl, peptide bond. So this basically is the primary structure, and as you see in orange, these are the side chains that they are really important for the next structure that is called the secondary structure. In the secondary structure, we have two common types. One is the alpha helix, and this looks like a corkscrew or spiral staircase where the carbonyl forms hydrogen bond with the nitrogen hydrogen bond from another amino acid in the chain. And the beta sheets are two or more pleated regions looking like the shape of a pleated skirt or corrugated cardboard, are held together by the same H bonds as in the alpha helix. One major difference is how far apart from each other the amino acids are in the amino acid sequence. Alpha helix atoms are much closer together. So here we have an example. Here is the carbon of a carbon. Remember that the red ones are the the oxygen, of the carbonyl, and the white is the hydrogen. So here we have a hydrogen bond, okay, between this oxygen that is of this amino acid with this hydrogen that is of this amino acid. So here we can see the interaction of the hydrogen bond between amino acid to create this type of structure that is known as the alpha helix. The other structure is the beta sheet, and we can see here the same concept between oxygen and hydrogen and oxygen, between different amino acids from other, not the one that is closest to them as in sequence. So they can create this type of arrangement that is called the beta sheet. So the protein structure, the secondary structures, alpha helix and beta sheets are basically hydrogen bonds, okay, created between the carbonyl group and the hydrogen of different amino acids of the primary structure. The tertiary structures are basically side chains interaction that interact with each other and the surrounding environment, usually aqueous environment. The forces we call intermoleculars are mostly what drive tertiary structures. So basically, the side chains have polar, nonpolar, and charged groups. So this gives rise to the ion-ion, ion-dipole, dipole-dipole, and dispersion force interactions. That is, are those forces, those intermolecular forces that can stabilize that type of structure. So here we have basically that the tertiary structure. Remember that are the interaction between the side chains of the secondary structure. We have here a alpha helix. These are the representation of beta sheets, and they can interact between them, okay, by the side chains. And if we have side chains with ion or dipoles, so we can create that type of interaction between them. The quaternary structure. Some proteins are made up of more than one polypeptide chain. Some proteins contain portions that are not amino acid in nature, and the combination of subunits is the one that create this quaternary structure. So basically, the combination of those tertiary structures and also sometimes with non-amino acid portions are called the quaternary structure. Okay. So now let's classify proteins. One method to classify proteins is based on the solubility. We have two types of protein. The first one is known as the globular proteins and are roughly spherical and dissolved in aqueous environment. The second one is called the fibrous proteins and are usually long fibers that are insoluble in water and are used as structural materials. So we have the globular one that dissolves in water and the fibrous protein that are insoluble in water. Let's talk now about carbohydrates. The name of carbohydrates comes from an empirical formula for sugar that has carbon and H2O. That they at the beginning they thought that it was carbon and water, so they called them carbohydrate because we're molecules that have water. But then we saw that basically they don't have water, even though the combination is H2O, the ratio, okay, but the molecule by itself doesn't have any molecule of water. But that's why they call carbohydrate, because at the beginning that empirical formula shows something like that, like CX H2OY. So simple sugars as monosaccharides are polyhydroxide aldehydes or ketones. They are often drawn as chains, but most frequently exist as rings in solution. So here we have the glucose, alpha glucose or beta glucose. So basically, this is with respect to this OH with the ethanol here. So this is where they are opposite sizes, alpha, and when they are the same side is beta glucose. Monosaccharides. The two most common monosaccharides are glucose and fructose. Glucose is an aldehyde. Here we have the carbonyl group with one hydrogen. Remember that if a carbonyl group has one hydrogen, that is called an aldehyde. And here we have the fructose, that is a ketone. We have the carbonyl group and both substituents are carbons or alkyl groups. Okay, so that's why we have here a ketone. This is the fructose. The fructose is a ketone and the glucose is an aldehyde. We also have disaccharides. The hydration between two monosaccharides form a disaccharide. Sucrose and lactose are both disaccharides. Here we have sucrose, and sucrose is the combination between a glucose unit and a fructose unit. They combine together, creating a disaccharide that is called as sucrose. And lactose is a combination between galactose and glucose, and this create the lactose that is another disaccharide. The saccharides are often referred also as sugars. And we have the polysaccharides. The polysaccharides basically are dehydration can create long chain polysaccharides. The three most commons are starch, glycogen, and cellulose. These are the three most common polysaccharides. The starch consists of many different sized and various branching chains of glucose prepared by plants to store energy. The glycogen is often called as animal starch. It is for temporary energy storage in animals which use fats or for longer term energy storage. And the third one is the cellulose, that is a structural polysaccharides in plants making up cell walls, and it is unbranched. So here we have an example at the top one. This one is the starch, and in the bottom one we have an example of the cellulose. Now let's talk about the lipids. The lipids are a broad class of nonpolar bioorganic molecules. They are grouped because they are insoluble in water. All the lipids are insoluble in water. They are used biologically to store energy, like fats or oils, and for biological structure, like phospholipids and in cell membranes. So let's talk about fats and oils. These are made from long-chain carboxylic acids and glycerol. They have only saturated carboxylic acid, so that means that they don't have any type of double carbon double bond or triple bond. They are solids. These are often called the bad fats in your diets. Oil Oils have at least one unsaturated carboxylic acid. They are liquid. That's why they are liquid, because they have that unsaturated unit between by due to those carbon double bond or triple bond. The more unsaturated, the better for you. The polyunsaturated versus monounsaturated fats. And the essential fatty acids with double bonds must be included in our diet. Our bodies can't produce them. They are often called of omega-3 and omega-6 fatty acid, and you can find them commonly basically in fish. So the phospholipids, um, how their structure is similar to fats, glycerol with ester linkage two fatty acids instead of three. How it differs: the third side has a phosphate ester linkage connected to a charge or polar group, such as choline. They cluster together in water, polar regions pointing toward water, non-polar fatty acid regions pointing toward each other, forming a lipid bilayer, the start of a cell membrane. So here we have a group of phospholipids. The blue one, the blue circle basically is the hydrophilic portion of that phospholipid. And basically those yellow legs are the lipid or the fatty acid region, the non-polar. Okay, they interact between them, creating this bipolar lipid bilayer. Sorry, the lipid bilayer. Okay, that create the cell membrane. So this one is hydrophilic, this one is hydrophilic, and the middle part is hydrophobic. Okay, so basically these are those phospholipids, and they are really important. Okay, because for example here we have this glycerol. Okay, this is the comparing the phospholipid to fats. So we have the glycerol here, these are from fat, and we have here the phospholipid that has the phosphate group here, and you can see here that this is the hydrophobic tail, but some of them can create this type of bend because of the presence of this double bond. This is unsaturated fatty acids, okay, the ones that are um, uh, the one that we need in the body. They are better than the ones that are basically saturated. Okay, so this will a large amount of this one can affect us in our daily way. So finally, let's talk about the nucleic acids. Nucleic acid, this is a class of biopolymers that are chemical carriers of genetic information. We have two types. We have the deoxyribonucleic acid, or DNA, and the ribonucleic acid, the one that is called as RNA. The DNA basically is a huge molecule, 6 to 16 million MAU. Primarily is found in the nucleus of the cell, and they are really important because they stores genetic information and also specifies which proteins the cells can synthesize. So the DNA is basically is like the instruction book to create proteins in our body. The RNA, they are smaller molecules, smaller than DNA. They are from the size goes from 20,000 to 40,000 atomic mass unit, and mostly you can find them outside of the nucleus in the cytoplasm. They carry stored info from DNA into the cytoplasm, and the information is used in protein synthesis outside of the nucleus. So these are also really important. They can also help in the regulation, basically, of the production of protein. Okay, so the DNA basically is the ones that give the instructions, and the RNA sometimes helps also in the regulation of the synthesis of protein. So what about the structure of the nucleic acids? This consists in three parts. The first one is a 5-carbon sugar, also known as the ribose or deoxyribose, a phosphate group, and an N-containing base. These are called as adenine, guanine, cytosine, and thymine, or uracil. So we have 5 different bases that can be used to create this DNA. And the polynucleotides form by condensation reaction between a phosphate and a sugared OH. Okay, so we have here a phosphate group. Here we have an example of one complete base here. We have the 5-carbon sugar. Also, we have the phosphate group here, and we have the N-containing base unit. And basically the combination between them create this phosphate bond, okay, between those bases. And this is the polynucleotide that can create those interactions to produce the double helix of the DNA. So here, in the nucleotides combine to form the familiar double helix form of the nucleic acid. And these are formed because of the H bonding and the dipole-dipole interaction and dispersion forces that hold together the double helix. Okay, help to keep them together closer. So the complementary base pairs form ideal H bonding partners between adenine and thymine. These are basically double bonded, and the triple bond between cytosine and guanine. So here we have here basically the DNA, and you can see the G, the A, the G for guanine, C the cytosine, A the adenine, and T the thymine. And there is the interaction between those bases that produce this type of helix form between the DNA. And as you can see, the thymine and the adenine, the interaction creates 2 hydrogen bonds, while the cytosine and the guanine create 3 hydrogen bonds between them. And this is the interaction that gives stability to this type of double helix. So with this, we'll finish our module 24 of the chemistry of life, organic and biological chemistry.